Hello, everyone. Welcome to today's live broadcast of a bioanalytical discovery strategy for fast and flexible decision-making. I'm Jeannie Linky-Northrup, Managing Editor with Pharmaceutical Technology, and I'll be your moderator for today's event. In today's webcast, we are joined by Ryan Adler and Edward Brewer, both of Illyri Bioanalysis. Ryan and Edward will react to advancements in the field and share some strategies that can be employed to help streamline your workflows and advance molecules with ease and precision. Today's webcast is sponsored by Illyri, renowned for its role as a leading bioanalysis contract research organization with locations in Colorado Springs, Salt Lake City, as well as across in Luz, France. Illyri offers a range of bioanalytical lab, spatial bioanalysis, and spatial biology solutions to pharmaceutical companies. The company provides assistance to navigate through all stages of drug development, and the team's industry-leading science and regulatory compliance expertise makes Illyri a key player when advancing molecules from discovery through clinical development and NDA approval. Before we begin with today's webcast, there are a few important announcements I'd like to make real quick. First and foremost, this webcast is designed to be an interactive discussion, and so we encourage you to ask any questions you may have to our presenters throughout the event. You can submit your questions by typing them in the Q&A box that can be found at the bottom of your video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of your media player. Please note, though, however, slides will advance automatically throughout the event. If you do experience any technical problems viewing or hearing this presentation, please click on the question mark help widget that's located in the top right of your presentation window. Now I'd like to further introduce today's experts and guest presenters. Ryan currently serves as the manager of lab operations for Illyri in Salt Lake City, where he oversees the sample analysis, validation, discovery, and quality control teams. Throughout his career, Ryan has held many positions, including roles in lab operations, discovery, method development. He is a previous principal investigator, and he has extensive experience with all study types from early phase discovery through clinical research. Ryan has worked with multiple matrices, including a large variety of tissues. Edward joins us today with extensive experience in bioanalytical, mass spec, dating all the way back to 1986. He currently leads Illyri's non-GLP programs. His expertise comes from experience with forensic toxicology, large pharmaceutical companies, and contract research organizations. Ed's research interests involve quantitation of pharmaceutical-based products from biological matrices with high throughput assays. So gentlemen, welcome. Thank you so much for joining us today. Feel free to go ahead and get started with your presentation.
Great. Thank you so much, Jeannie, for the introduction. I will get the presentation up on the screen here. All right. Thank you, Jeannie. As indicated, my colleague Ed and I will be talking to you today about a bioanalytical discovery strategy for fast and flexible decision-making. We'll start out with an introduction to discovery work at Illyri. Many of us should be familiar with this image, which is the typical drug development timeline. Drug development typically starts with basic research, moves into the early discovery phase and hit-to-lead screening, then into preclinical work. Hopefully from there, we move into filing an IND application. We move through phase 1 through phase 3 of clinical trials, submit an NDA application, and we all hope to then have the FDA review leading to approval. Today's talk will focus mainly on this section here, the early discovery phase of drug development. First thing to be aware of with discovery is that it is fit for purpose. Whenever I'm discussing discovery with either sponsors or internally, I always emphasize the fact that we need to really understand what the purpose of the study is, what the needs of the sponsor are, in order to understand what type of assay we'll be developing to support that study. Questions that we ask initially to figure out the type of study is what stage of drug development are we in here? Is this early phase screening? Are we really focusing on that hit to lead Have we already identified that lead candidate and we're working on lead candidate optimization? Is the lead candidate already fully optimized and we're now preparing to move into those preclinical trials and GLP work? What type of matrices are we working with? Are we working with plasma, urine, maybe some other biofluids? Is there tissue involved? After these basic questions are answered, we can start to determine what assay tier this should be a part of. At Illyri, we use a tiered system for determining the assay type that we're going to develop and what goes into that assay. So do you need a rapid turnaround of data, less than 72 hours, using a preliminary method that has a wider acceptance criteria? That's something that we would consider a tier 1 assay. Do we need tighter acceptance criteria, a more optimized method where we're really looking forward towards that GLP work and hoping to use the method development that we've put into this discovery method to help support a GLP method down the road? This is something that would go into a tier 2 type assay for us. Or are we near GLP? Do we maybe need to fully develop a method that is ready to be GLP validated? However, maybe we are going to be working with some non-regulated studies. We don't necessarily have the time to validate. We want to get that last bit of data before we move into GLP work. There, we're going to be in what we consider a tier 3 assay. Let's talk a little bit about what type of studies go into those tiers. So for tier 1, we are focusing on studies that are lead generation type studies, hit to lead screening. It's the stage in early drug discovery where chemical hits from high throughput screening are evaluated to help identify the lead compounds. Are we in lead identification with chemical compounds that have pharmacological or biological activity that we expect to be therapeutic? They can be agonist or antagonist to a receptor of interest in a small molecule type program, or they could be up or down regulating in an oligonucleotide type program. For tier 2, we're really into our lead optimization. We're looking to improve potency and selectivity and pharmacokinetic parameters by the evaluation of structure-activity relationships. Tier 3 are those near GLP studies. Maybe we have some non-regulated studies. PK/PD studies can be non-regulated. We're trying to get that last bit of data as indicated before we move into GLP work. Now, when we're working with various biofluids, they can be categorized into tier 1, tier 2, or tier 3, depending on what stage we're in. If we're working with tissues, we've got tier 1 and tier 2 is most common. However, if we're working with a target tissue, then we would consider placing that in tier 3. Again, discovery is fit for purpose. That's something that we work with the sponsor to determine what is appropriate for the assay. Let's talk a little bit about what goes into the criteria and the design of these different tiered assays. For tier 1, we're really focusing on that yes-no decision-making, or you could say the go, no-go decision-making. Is this going to be a viable candidate? For that, we may be using a generic protein precipitation method and maybe a steep gradient on the LC in order to provide rapid turnaround of data. There's minimal method development that goes into that in order to get that data out quickly. We're going to have accuracy and precision at plus or minus 25% for biofluids. However, we do add 5% to that for plus or minus 30% for tissue matrices. We will use a single weighing. We will prepare calibrators. However, depending on the sponsor needs, we may add QCs to that as well. We will go through some level, minimal level of method development, maybe just tuning up the compound, ensuring we have appropriate chromatography, before employing a kind of a standard method. We will go through a method qualification, although at times, again, with that fast turnaround, if we're working with a class of compounds where we know a certain method type is proven to work, we may forego that qualification, move straight into the sample analysis in order to turn that data around in as short as 48 hours. All of the data will be fully PI reviewed before delivery to the sponsor. Moving into a tier 2 assay, a lead candidate optimization type study, we're going to tighten up our accuracy and precision criteria to plus or minus 20%. Again, for tissue matrices, you add 5% to that. We are still using a single weighing. We will prepare calibrators and QCs. We'll do a bit more extensive method development. We are starting to look forward to those GLP studies. We want to be able to use whatever method development we've put into developing this level of discovery assay to benefit us for when we get into the GLP work, and hope to condense the time it takes to develop a GLP assay. We'll do a qualification run. At times, we will add stability testing into this. Again, fit for purpose. We need to have that discussion with the sponsor to understand if any stability or what stability might be required at this stage. We'll do our sample analysis, and again, the data gets a full PI review. When we move into tier 3, this is near GLP. Ultimately, there is no QA oversight of a tier 3 discovery assay. For all intents and purposes, everything is done as close to GLP as possible. But since the studies are non-regulated, we do not have QA oversight. Accuracy and precision will be at plus or minus 15%, which is identical to what would be for a regulated study. Again, for tissue matrices, we add 5% to that. We do, at this point, do duplicate weighings, and we prove accuracy between those weighings. Then we prepare our calibrators and QCs. We will have more significant method development. We would expect that a method that is in a tier 3 status would be very close, with maybe only minor tweaks, to be able to be validated for a GLP assay. We'll do a method qualification. We will likely do stability testing. We'll run our sample analysis. And now, instead of just having PI review of all of the data, we will also have full QC review. Let's talk a bit about matrix considerations. In discovery, we can run all types of matrices. They can be categorized into the 2 categories here, biofluids or tissues. Biofluids, as I indicated before, can be run at any tier of discovery, and many of them, pretty much all of them, can be validated. Tissues, on the other hand, will always be discovery. It's very rare that we will try to validate a tissue assay. The main reason for that is stability testing. We cannot establish stability in a tissue matrix that truly mimics the stability that you would experience of the analyte in that native tissue as it's dosed into the body. The reason for that is, in order to analyze a tissue sample, we do need to homogenize that tissue prior to being able to aliquot it. So we would first homogenize the tissue. We're going to then spike analyte on top of that homogenate, and that's how we would test stability. That does not represent what is happening in the native tissue, so therefore these assays are typically not validated. Some of the tissues will need to go beyond homogenization and will have to be digested as well. Skin, for example, is a very tough tissue to work with, and we would need to digest that in order to fully homogenize that tissue. Another thing to be aware of when dealing with things like vitreous humor. Vitreous humor can be a biofluid. It can sometimes be aliquoted out. but sometimes the samples we receive are more gelatinous, and we would also need to homogenize that tissue. Speaking of homogenization, we should talk about the homogenization techniques that we employ at Illyri. The first technique, which is the least used of the three techniques I'll talk about, is mechanical. Mechanical uses a handheld probe for homogenization. It is an extremely slow process. You will only be homogenizing a single sample at a time. You're using a probe that has a blade embedded in it. You insert that probe into the sample tube. Sample tube has homogenization buffer added to it, and that blade will mechanically homogenize that sample. Between each sample, you will need to wash that probe thoroughly to ensure that you've gotten out all tissue particulates and any drug that has stuck to that probe tip. So that thorough washing, again, will take time between each sample, and you're prone to contamination as you can have tissue particulates that end up in that blade that you can't necessarily see. So you do have to make sure you're being extremely thorough when you are cleaning that probe. The benefits here are if we have a very large tissue, this is an approach we could take to deal with that. Bead beading is very common, especially in small molecule work. Bead beading uses beads of various materials and sizes. Very common would be silica zirconia, or potentially, if you want to go on the other end of hardness, a stainless steel bead. We can use beads of 1 millimeter, 3 millimeter, or even larger, depending on the hardness of that tissue. Once you add your homogenizing buffer and your beads to the sample vial, you then put it into the bead beater, it shakes vigorously, and you end up with a homogenate. The final type that we use is powdering. Instead of adding a homogenate or a homogenizing buffer to the sample prior to bead beading it, here we're going to take that dry tissue. We're going to add usually one large stainless steel bead to it. We're going to put that into the equipment under liquid nitrogen so we maintain a frozen state, and we will turn that tissue into a homogeneous powder. There are a lot of benefits to this. We can take aliquots of that powder, which is already homogeneous, and we can homogenize a portion of it, leaving behind the rest of that tissue for future use, whether that's going to be for reanalysis later and that tissue technically hasn't been touched beyond powdering, or maybe the sponsor needs that tissue back for doing other types of work. If we've already homogenized the sample, it may not be in a state that it can be used for other testing that we're not doing here. However, at a powder, it can be put into whatever state it's needed for that further testing. So working with tissue, when we're developing assays, there are a few different approaches that we commonly take at Illyri in order to analyze our samples. First one is a matrix matched approach. This is least commonly used, although when we are working with the target tissue, at times in a tier 3 assay, we will take this approach. This is where we're going to prepare calibrators and QCs in the same tissue matrix that the study samples are in. This can be very costly, as blank tissue matrix is expensive, and it can sometimes be hard to come by depending on what types of tissue we are working with. It is slower, as we're only able to analyze one tissue type per preparation. If your study has multiple types of tissue, we would need to make pools for each of those tissue types. And again, the cost increases there as well for that blank matrix. A more common approach that we take is what we consider the spike on top method, We will prepare neat solutions, essentially a calibration curve and QCs in neat solution. We will spike that directly on top of a blank matrix aliquot in our sample plate so that the final concentration in that plate is the concentration that we want our calibrators and our QCs to be. The benefit of this is we are using that minimal amount of blank matrix. It is still matrix matched when we do that. It brings that cost of the blank matrix way down. Instead of preparing pools of multiple milliliters, we're using, maybe a 50 microliter aliquot for each calibrator and QC in a run. We are essentially doing a fresh preparation daily when we do this, as we are spiking those neat solutions on top fresh in each run. However, the neat solutions have been verified prior to use as linear and accurate. The most common approach we take is what we refer to as a 50-50 approach. This is where we will prepare a plasma curve as the most common. Although when we do have a target tissue, sometimes, again, discussion with the sponsor, we decide to use that target tissue as that initial curve in place of the plasma. But if we're using a plasma curve, we can prepare and qualify a plasma curve. The matrix cost is minimal compared to blank tissue. We will aliquot out that plasma curve and QCs into the run plate. We will aliquot our unknown tissue samples into the plate. We will then take an equivalent aliquot of blank plasma and add it on top of every unknown tissue sample in the run. We will take blank tissue matrix matching the unknown samples and take an equivalent aliquot and add that on top of the plasma curve and QCs. What that effectively does is it makes every sample in that plate, as far as matrix is concerned, 50-50 plasma tissue. Your matrix effects are now normalized throughout the plate, so you should have no impact from using a plasma curve to analyze your tissue samples. By doing this, we also have the ability to combine multiple matrices into a single batch. We can have a set of QCs representing each different tissue matrice in the run. If we've got 3 different tissue matrices, 3 different sets of tissue samples in the run, the QCs will will aliquot out plasma QCs into those 3 sets. Set 1 will get blank tissue from set 1 of the samples. QC set 2 will get blank tissue from the second set of samples, and similarly for the third set of QCs. As long as those QCs match the calibration curve and everything proves accurate, you know that there are no matrix effects that impacted one set of unknown samples or one matrice differently than the rest of the run. If they meet acceptance criteria, you can accept the entirety of that run. One of the challenges that we deal with when working with tissue samples is the unknown distribution throughout tissue. The ideal situation is that we are provided the entire tissue in order to homogenize that so that we are representing the entire tissue when analyzing the data. When we have had issues where maybe a sponsor did not get the data they were expecting, maybe they sent us a portion of the tissue, maybe they sent us a region of that tissue that didn't have any drug absorbed into it. Maybe they sent us a region that had all of the drug absorbed and the concentrations were way higher than expected. At our Aliri, France site, they have the ability to help when these issues arise by doing spatial analysis. What they're able to do is image that tissue and help elucidate some of these tissue distribution issues. What I have on this slide is an example of that work where epicadostat is used to inhibit the conversion of tryptophan to kynurenine. You can see in the bottom row of the images, the epicadostat anneals or binds preferentially to a specific region of the tissue in the upper right-hand portion. Tryptophan is going to be similar in both the control and treated. However, you do get preferential inhibition of that conversion to kynurenine in the treated group in the region where the drug bound. We have had sponsors that have gotten data that didn't quite make sense, and it came down to tissue distribution issues. They have been working with our team in France to help elucidate some of those issues. I will now hand it over to Ed to discuss small molecule work specifically.
All right, thank you, Ryan, for that introduction. Next slide. I just want to go over a few of the programs we have supported with discovery, and mainly we're an LC-MS shop, so we do a lot of quantitation, and point out that the tiers that Ryan we're talking about is just a starting point for conversations, and our team member would certainly be able to help support whatever your needs are moving through that. So, basically a single molecule, a single rodent study is a typical program for discovery work. The other things we can support are cassette dosing, where you take a single animal series and dose them with multiple analytes. That provides information on multiple analytes. And, we're using the advantage of the mass spectrometer to differentiate those compounds by weight. One of the things you want to be cautious about are isobaric and isometric interferences for those. You typically want the molecular weight of the compounds to be at least 4 AMU apart. And analogous to that, you can also do a single dose to a single animal, and we can combine those so that we save on the back end for the bioanalytical assay, knowing that the samples are getting diluted. We want to make sure that we keep the lower limit of quantitation effective still. So, instead of running our typical 1 to 100, 1 to 1,000 nanogram per ml, we may be looking at 0.1 to 100 nanogram per ml, typically 3 orders of magnitude on the quantitation line. And next slide, please. We can also support libraries. Here we take a generic methods developed on a single molecule for the class of new chemical entities, and we develop a method and qualify that method. And any additional compounds in that library are run at risk against that method, assuming that the method is going to work for all compounds in that library. So that's an easier way to screen a library without having to develop a method and qualify a method for each and every compound in the library. Next slide, please. We also have software options that can help support our quantitations. If we know that there is a metabolite present, like an N-desmethyl or an N-des, and you want to find out what that is what the quantity of that is without going through an actual production of the compound, we have the ability within our Watson limb system to do a regression against the parent molecule. And what that requires is that, the caveat to that is the ionization efficiency for the metabolite and the parent are pretty close. But it does give us an idea of if the metabolite is worth going after or not, and spending the time and money to making the actual metabolite. We also have the ability within the Watson limb system to provide basic PK parameters like area under the curve, clearance, half-life, for studies that are non-GLP based. And that's just a matter of talking with your O'Leary team member and getting that sorted out. And we have the capability for send PC domain data, which is available from our LIMS system. Have the next slide, sir. One of the questions we get asked quite often is, what is our turnaround times? It's typically about a week from about 5 business development days from the time that all the materials are here to provide the results to you in an Excel format. We also have the option to speed that up if there's proper communication between the client and the staff member, so that we can actually run the samples the same day that they are received on site, typically by 10 a.m. We can have those on the instrument and that evening and data the next day. Some of the things that we look at during that are the samples run at risk with the qualification run. Tissues are matched 1-to-1 with plasma, as Ryan spoke earlier. And we usually use an analog NCE or an IS cocktail for the internal standard. Right. Next slide, please. I'm going to turn this back to Ryan now to talk about oligonucleotides.
Thank you, Ed. So moving on to some specifics on oligonucleotide discovery work at Illyri. First, just to show some of the typical structure and modifications of common oligonucleotides that we work with here, we have various conjugates like GalNAc conjugates. We have sugar modifications. We've worked with base modifications, phosphate modifications, sugar phosphate modifications, including PMOs and PPMOs. When dealing with small molecule work, we're really focusing our instrumentation on triple quad mass spec being the most common. However, with oligonucleotides, we do have 3 different platforms that we employ. One of those, and one of the least frequently employed, is LCFD. The reason that this is least frequently employed is that we have the benefit of lower LLOQs than our other platforms. However, typically we are looking for higher concentrations in discovery work of the oligonucleotides. If you're looking for very low concentrations, then we would propose using LCFD. Specificity on LCFD increases with the size of your oligo. We do have extremely long run times when we are doing FD work. We're typically in a 20 to 30 minute range per sample for LCFD. Another challenge with LCFD is dealing with the separation of the metabolites, the N and N-1, N-2, and so on. Metabolites can create a challenge as you don't get specificity from those by the method of detection. We will need to work on that separation. However, if the sponsor does not have those reference materials available, it is an unknown, and chromatography that we have when we qualify our method that is perfectly symmetrical could look more like a fingerprint region once you have all the metabolites in your study samples. LC-MS-MS, or triple quad mass spec, is more commonly used. LLOQs are around 10 nanograms per mil. We do get our selectivity through fragmentation, and we have the benefit here that our run times are more on the order of 5 to 8 minutes rather than the 20 to 30 minutes that we have for LCFD. Most common for the various oligos we work with is high-res accurate mass spectrometry. Again, being mass spec, our LLOQs are similar to a triple quad, around 10 nanogram per mil. Our selectivity now, however, can be based on the accurate mass of that fragment or of the intact oligo, which can provide a lot of benefit. We are able to look at various charge states. We have a much larger mass range than we do on a triple quad to enable that analysis. Run times are similar to triple quad, but again, much faster than LCFD. We also can run in full scan analysis mode, which enables further data mining. We can provide that data back to the sponsor to enable them to mine for various metabolites. With oligo work, we should always talk about throughput. Oligos are by nature more complicated than small molecule. While small molecule turnaround times can be as short as 48 hours, oligo turnaround times are typically longer. Depends on what's going into the study as to what those turnaround times are. Major reasons for that are method development challenges. When dealing with oligonucleotides, we commonly deal with protein binding issues. We deal with absorption issues. We deal with carryover issues, carryover in the LC system, in the syringe, the injection valve, the needle, or on the analytical column. The columns that are typically used for oligonucleotide are also prone to excessive carryover. So we need to test for all of these things and work out those issues so we can assure that we are providing accurate data. The extractions themselves are lengthier. When we're doing a solid phase extraction for a small molecule, it's a fairly quick extraction. For oligonucleotides, there are more steps. We typically employ more washes, maybe additional elutions in order to ensure we're optimizing our recovery. Our final sample is usually highly aqueous. In order to hit the LLOQs we need to concentrate that sample. Because it's highly aqueous, the evaporation times can be on the order of a couple hours. Mass spec run times are longer than a small molecule assay. Small molecules, we commonly run for discovery 2 to 4 or 5 minutes on your LC, whereas you're more on the 6 to 8 minutes per sample for an oligonucleotide. As mentioned earlier, if you're using fluorescence detection, you do need to account for the truncated forms in your separation if we have the ability to, and the extensive run times. In order to help combat the longer turnaround times, we do have the potential, as I discussed, to combine especially tissue matrices into a single run by that 50-50 method to significantly improve throughput. If you have a study that is going to involve 8 different tissues, doing that as 8 separate runs versus maybe doing that as 2 or 3 runs will significantly cut down on the extraction time as well as the analysis time, which will directly impact positively your turnaround time. Couple things we need to keep in mind for tissue analysis. With oligonucleotides, homogenization typically includes digestion. This helps free the oligo from bound proteins. You do end up with a much cleaner homogenate. That means that your instrumentation is going to go smoother. It also keeps your internal instrument engineers much happier with you when you're injecting cleaner samples on a daily basis. Be aware of what you are working with. It is not okay to digest PPMO as it would be prone to cleavage. Homogenization does typically dilute the sample around 10x. We can and do optimize that dilution volume dependent on the needs of the study. If you provide a very small tissue, maybe a mouse tissue, we may need to up that dilution factor in order to have enough volume to be able to aliquot, which will dilute your sample further. On the other hand, at times we can dilute less than 10x and still get a homogenate, but we need to make sure that we're paying attention to the solubility of the analytes and using a homogenizing buffer that addresses the solubility, as well as using enough homogenizing buffer to fully solubilize and fully homogenize this sample. When thinking about that dilution factor, it's important to realize what we're talking about here. So a 10 nanogram per mil LLOQ is 10 nanograms per mil of homogenative. When you back calculate that into the native tissue, accounting for the weight of the tissue, that is 100 nanogram per gram in tissue. It is always best to work with the entire tissue when possible to avoid any tissue distribution variability. As I talked briefly about earlier, our team at O'Leary, France can help you out with spatial analysis if you have any potential questions or concerns with your tissue distribution of your drug. For oligonucleotides, it is very common for tissue work to be attached to a GLP protocol. With that, the tissue portion is accepted from the regulatory requirements, and we will typically analyze the tissues as tier 1 or tier 2, most commonly tier 2, when attached to that GLP protocol. However, if it is the target tissue, then we will consider running that as a tier 3 assay. Again, all of these types of conversations occur with the sponsor up front, so we are all on the same page of what the needs are of the study. I will turn it back over to Ed one more time to talk about proteins and conclude us today.
All right, thanks, Ryan. Now, we've talked about small molecules and oligonucleotides. I'd like to take just a minute to mention about our work with proteins. Next slide, please. Typically, we're looking for a human version of a protein in a non-human species. That could be like, we're looking at a human version in a monkey species. So as long as there's an amino acid difference between the human and the monkey, we can pretty much separate that on LC-MS. The first thing we would do in this case is run an in silico review of the proteins and tryptic cleavages. We would identify peptides via a time of flight that we have here in Colorado Springs. And then we go ahead and purchase the peptides. We have them prepared and purchase those along with an internal standard. And that usually takes 3 to 4 weeks from our vendor. We would go ahead and develop the method at that point, isolate the signature peptides. And this is typically a 20-minute LC-MS method to get that separated and identified with LOQs around 10 to 100 nanograms per ml. I'd like to point out that we have a protein webinar coming up at the end of the month, and the link to that webinar is on the screen at this time. Next slide, please. So in concluding our talk today on small molecules and discovery work, next slide, please. Data is provided from our Watson LIMS system in an Excel format, so there is no remanipulation of the data. It's strictly from our Watson LIMS system, and all programs go through that. We can have PC-Send data available from the LIMS system, and abbreviated reports are available upon request. We do have the ability to perform PK information on that system if that's of benefit. And the turnaround times can be as short as 24 to 48 hours. Typically, everything is done within 5 business days, and that just requires prior communication with your O'Leary team member and running a pre-established method, or are we running the samples and the method qualification at risk, assuming everything's going to work out okay. And that's usually performed when we know we have enough matrix to go back and run the assay again should something go wrong. And then we have quantitative information available for a wide variety of experimental designs. We would include protein binding in that as well, either through the RED device or through a molecular weight cut filter. Next slide, please. So this time I'd like to thank you for your attention. Ryan and I will be available to answer any questions you should have. And again, we have sites in Salt Lake City, Utah, Colorado Springs, Colorado, and Little France that Ryan has mentioned it before. And thank you for your attention.
Great. Thank you so much, Edward. And Ryan, thank you so much for all of your insight as well today. I have seen some really great questions starting to populate in from the audience. And for anyone who may have joined a bit late, I want to remind our audience that this is an interactive discussion. And at this time, we're going to get ready to open up the floor for our Q&A session. So if you have a question for Ryan or Ed, you can submit it through the Q&A box that can be found below your video player. So gentlemen, as we start to explore some of the questions coming in, the first question that I have, okay, I know tissue is the issue, right? That's what they say in healthcare, especially. And turnaround times are very, 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 very important and becoming even more important as we talk about daily in this field. So is there anything that can be done in terms of what the client can do to help facilitate the tissue analysis and reducing the turnaround time?
Yeah, absolutely. So we typically work with our sponsors to provide them information that can help in this regard. So one of the time-consuming pieces to tissue work is we do need to get the weights of each individual tissue before we homogenize so that we can add the appropriate amount of homogenization buffer to those samples and dilute them at the proper level and be able to calculate back accurate concentration data. If we receive tissues that are already pre-weighed in the tubes that we can homogenize in, that won't break upon that violent shaking, then we can move straight into that homogenization process in order to cut down on that front end. If we need to weigh the tissues out, if we're getting 50 tissues, if we get 400 tissues, that can be a huge deal. So we're looking at the savings of potentially a day or two to as much as a week when we get a large volume of tissues in. We do commonly provide part numbers for the tubes that we can use for homogenization, depending on the size of the sample, so that they can be provided in that manner. Of course, if that's not feasible, we can do all of that front-end work. It will just take a little bit of extra time.
Great, thank you.
I would just like to reiterate what Ryan's saying, that if the tissues come in pre-weighed, then we can generally do a quicker turnaround than taking an extra day to weigh them and homogenize them. So that really does speed up our analysis on tissues.
Thank you, Ed. That's what I was going to ask you if you had anything else to add to what Ryan had to say in terms of some of the capabilities through the equipment, for sure. Another question that came in that will probably, we're very quickly approaching our hour, but we'll probably wrap up our discussion. How are decisions made when the data is not expected? You know, I know Ed specifically was highlighting some of the data and the capabilities through the different equipment here, but what happens if something's out of range or anomalous data?
Yeah, that's typically a conversation with the sponsor. It's typical to provide extrapolated values for ULOQ samples so that they have an idea of what the value would be on a linear fit. But typically those are repeated with dilution to be in range to the assay.
Yeah, to add to that as well, with the ULOQ samples, as Ed mentioned, with a linear fit, with a linear regression model, there is accuracy when we provide extrapolated data above the range. That is very common, especially in some of those earlier tiers, lower tier assays. We have seen time and again when we reanalyze those samples with dilution, so they are in range, that the result we receive matches what the extrapolated value was from the original. Below LLOQ samples, again, discussion with sponsor. Sometimes they're looking for, hey, do you see anything? Maybe it's below the limit of quantitation, but can you detect that there's drug there? We won't quantitate that as it's below the range, but we can say that there is positive value there. If it becomes important to have that data, then we can discuss developing an alternate range to be able to rapidly reanalyze those. So develop the lower range and qualify that range. With things like maybe IS responses that are out of spec or carryover that could potentially be impactful, we can take a scientific look at the data. That is the key to discovery work, is we really can focus on the science, whereas GLP is a lot about the compliance aspect. We can look at the data scientifically, and we can see, maybe it's a biofluid that has a PK profile. We can look at that PK profile from the rest of the data, see if the data we got for that anomalous point makes sense with the rest of the data set, and again, discuss with the sponsor whether we should accept that as is or potentially reanalyze that sample.
Thank you. And that's something I had just a question on was, the capabilities to resample data and even capabilities to consider the potential to even resample tissue if that's possible. Ed, I know you highlighted 3 different techniques and you were able to suggest which technique is most beneficial in each situation based on proteins, oligonucleotides, other molecules of that nature. And I know you were highlighting some of the pros and the cons and the differences between the triple quad methods versus non. Was there anything else you would like to add just to suggesting what you see in practice in terms of analysis and utilizing each approach?
We can typically push the LLQ down if we go ahead in some ions. It's a little easier on a Orbitrap or a TOF instrument, any of the high-res instruments. With the triple quads, we need to run an additional transition to get that summation for a lower LLQ. But that would typically be able to raise our LLQs for those tissue samples. And typically, once we do the homogenization, there is sufficient homogenate to go back and run the assay again if we need to.
Great, good to know. Well, Ed, Ryan, thank you again so much for your time today. West Coast meets East Coast. It was a really great discussion. I do see that we've had some other questions coming in from the audience that we unfortunately did not have time for today. However, I want to let our audience know that both Ryan and Ed will be reviewing and responding to your questions offline since we are approaching our hour. So again, thank you. I'd also like to thank Leary Bioanalysis for making today's webcast possible. We hope everyone found this to be educational, and I'd like to thank, obviously, our audience for taking some time out of their busy day as we approach Q4 to tune in and listen to Ryan and Ed's broadcast as well. As we do conclude, we're hoping that everyone would kindly participate in a brief survey. The survey will appear on your screen just after today's presentation has concluded, and your time and insight is really valuable and we appreciate it. You'll also receive an email alerting you when this webcast is available for replay, and we invite you to forward that announcement on to any colleagues who may find today's presentation useful and may have missed it today live. So thank you again to everyone. We'll see you next time. Goodbye.